thesis defense, of course, and as is traditional, he's going to start by telling us where it came from and what he's going to do next.
Okay, all right. Thanks everybody for coming to my defense, and today I'm going to talk about my research for the last 5 years, but before that I should tell you something about where I came from and my future plans. So I come from a village called Chakla in India, and it's very close to the border of Bangladesh. So my father, his family came from Bangladesh, and my mother's family is from West Bengal. And this is a picture of my village, and I think it's 2 winters back when I went home. And this is me at very young. And it's a very important photo of my life. And at that point, there were 3 members of our family, and now this is my sister. She's studying history, just finished her master's. And I'm from a big joint family, so we have like 14 members in our family. Pictures out there. So I did my primary schooling and high schooling in my near my village, and after finishing my high school, I went to Ramakrishna Mission Vidya Mandira, which is sort of like a monastery. These are the monks that are the person that took care of us, and we had other teachers too. And this is like the whole batch of my year. And these are some of my really close friends that I met there. We're still great friends. After finishing my bachelor study in Vidya Mandira, I went to IIT Kanpur. I made really great friends there too. This is after we won a cricket match there. And this is the people in our chemistry department. And after finishing masters from IIT Kanpur, I came to the YSR group and started my journey as a graduate student. And I'm still doing that until this defense, I guess. Now where am I going after that? So if I plot the distance that I traveled from my home for all my schoolings, and this is a log scale, you can see. So I finish PhD and I go for a postdoc, I have to go to like moon or something. So I couldn't do that if there is no room there, so I'm gonna stay with Jim for a while as a postdoc. So we'll see if there is a university opens up in March. But anyways, the journey with YSR Group was a pretty fun journey, and I'm gonna talk about my findings from the journey today. So this is the title of my thesis. It's Spatial Biology of Transcription and Translation in E. coli using Super Resolution Microscopy. And I'm quite sure that a lot of you are from chemistry, so therefore I'm going to first explain all the terms of this title and then go into the actual problem. So what spatial biology means is it refers to a very specific kind of organization of components of a biochemical process that sort of helps that process. So if you think of the cell division process of E. coli, there's this protein called FTSZ that normally is present as a filament. So you can think of a single protein as being this blue sphere. But during when the cell decides to divide, this protein forms a ring called a Z ring, and that then pinches and the cells divide. Also that ring recruits a lot of proteins that are important for the cells to divide. So therefore, this spatiotemporal organization of this protein, like in time of division, it will organize that way, is important for that process to happen. So that's a part of the spatial biology of E. coli cell division. So we're going to study these kind of processes. And when you study spatial biology, the kind of questions you ask is where are the components localized, and then you ask how do they get localized that way? What are the factors behind that? And you also ask what purpose does it serve for them to be localized that way? The two processes for which I'm going to study spatial biology are transcription and translation. In any biological system, the major process of like transferring information from that is the genetic code from DNA to the functions of proteins are sort of done in 2 steps. The first step is you transcribe the DNA, and the enzyme responsible for that is called RNA polymerase. In most organisms, it's a multi-subunit enzyme, and it sort of reads the code from DNA and makes a single-stranded mRNA. After the mRNA is being made, then another protein, which is called ribosome, it's actually a ribozyme, so I shouldn't call it an enzyme, but it makes a protein out of that mRNA. And so these are the 2 processes for which we're going to study the spatial biology. So therefore, to study, as I mentioned, what we're going to look at is that where ribosomes and RNA polymerases are localized in the cell, what are the factors that may be responsible for that, and what function does it serve for them to be localized that way. And why are these kind of studies important? It's because you can think of this as like aeroplane, and And all the parts of these aeroplanes, of these systems, are sort of being carefully discovered and analyzed by geneticists and biochemists over the last 50 years or so. And so it's a very great wealth of data available for the components. But we are trying to sort of put together all the components and trying to see how the whole aeroplane works. So it's a step towards that kind of holistic picture to understand this biology. And the organism that you're going to study these processes is E. coli. This is one of the most studied organisms, and this is this is a picture of scanning electron micrograph of E. coli, and you can see that they're pretty well represented by a spherocylinder. So the rest of my talk, we're going to think of E. coli as a spherocylinder that has a rough length of 4 micron and a width of diameter of 1 micron. Now, why are we going to use super resolution microscopy for studying these processes in E. coli? The problem is the diffraction associated with any optical imaging. The proteins that we're going to study, their size is around 5 to 10 nanometer. However, if you image a single protein with a fluorescent setup, it looks like a big blob that has a diameter of around 500 nanometer. And so therefore, if you have two proteins that are pretty close together and you want to study their individual dynamics and distribution, it's very difficult because the blobs merge together and you cannot separate them. And so you can think of that and a case where the proteins are sort of localized in this kind of filaments inside the cell. And if the interfilament distance is less than this diffraction limit, which is sort of the half width of this blob, then you cannot see that filament, and they're going to be looking like this big blob. And so that's why we need somehow to break this diffraction barrier and look at structure and dynamics at very high resolution. Now to do that, one of the keys that if we can somehow separately look at those molecules, and because if we can look at one molecule at a time, we know the shape of that blob really well. It's roughly a 2D Gaussian. And so you can localize a lot of photons that sample that Gaussian, and you can get a very high precision estimate of the center of that Gaussian, which is close to the estimate of the position of the original molecule. Now, how do we separate them? Because in space we cannot do that because they're very close together. Now, we can separate them in time, and the idea to do that is to use these proteins called photoactivatable proteins. Here's a spectra of a protein called KEDA, and you can, when it's not activated, it gets excited by 480 nanometer light, and this green spectra is the emission spectra of that protein. Now, if I now activate that molecule with a small amount of 4 or 5 nanometer light, then it gets excited at 540 nanometer, and its spectra completely changes, and this is the new emission spectra. So you can think of that you have a bunch of proteins, and you give a very small amount of 4 or 5 nanometer light, so very few photons hit those proteins, and you get one of those molecules that gets activated. And now if I excite those molecules with 540 nanometer and look at somewhere in this region, I'm going to only see that molecule. So that's how you separate molecules temporally, even though they're spatially very close together. And that's basically the principle of super resolution microscopy. So if you remember, if you had a structure like this kind of filament, and because of diffraction limited imaging, it was really blurred, you could activate sparse set of molecules that are so separated that they don't affect each other's image, and you can localize their center to very high precision. In our case, it's going to be around 30 to 50 nanometer. And what you do is that you localize their centers and you bleach them, and then you store their positions and you activate some more states, some more of those proteins, and you localize their centers. And by repeating that process, you can get enough sampling to get those structures in very high precision. What you can also do is that if you're interested in dynamics, you can activate one of those molecules and follow its position over time to understand how it's moving inside the cell. So that's basically the principle of our work. Now, what are the goals? The goals are, we're going to use super resolution imaging to understand how RNA polymerase and ribosomes are distributed inside the cell. And also, using the super resolution based single molecule tracking, we're going to track the dynamics of RNAP and ribosome and see what we can learn about the system. Now, to do that, what we need to do is that the photoactivatable protein that I talked about, we have to put them on the actual proteins that we're going to study. And that's done by molecular biologists, and we have had help from outside our labs, our collaborators. Landic Lab and Gorse Lab has provided us strains where RNAP is labeled with a photoactivatable protein. Hee-Jun Choi in our lab has made a strain also on RNAP. I have had great collaborators outside our department. Julian Lab in UPenn has provided us with strains that have photoactivatable protein on this particular subunit, 2 of them actually, and one from Uppsala in Sweden, where we have a strain where the 58th subunit of ribosome is also there. So we're going to track all these things and look at their distributions. Now, we're going to study all these things in E. coli in a EZRDM, in a good growth media. This is EZRDM, the RDM stand for rich defined media. And so therefore the cells are happy there, and it's a clean media, so you can do single molecule imaging. Now what do we see? So all about a lot of preparation. And so first, when you look at the super resolution images of RNA polymers, what we first saw, these red outlines are sort of the boundaries of the cell bodies. And you can see that the protein does not fill up the whole cell. It's rather present as patches. And if you look at the white field images, which are low resolution image, but you can compare 2 colors. So this is signal from RNA polymerase that is not activated. And then this red patches, not showing very well, from the DNA binding dye that binds to the chromosomal DNA of that cell. It shows that the RNA polymerases are localized on those DNAs. And so therefore, you can see that RNA polymers almost don't go to the outer regions, and they're pretty well localized to the DNA. What happens when you look at ribosome? They're completely opposite. The ribosomes, we found that they are present in the end caps and in the region between the 2 DNA lobes. And so here are some super resolution image of ribosome positions. And these are white field images that shows you that the signal, the green signal appears where sort of the red signal does not appear. So ribosomes are very well segregated from DNA. Now, the super resolution images are quantitative images. So you can quantify the distribution profiles of these proteins inside the cell. So here's a single cell, and there's around 2,000 positions of ribosomes from that cell. And if we compare that from like the expected distribution, if the protein was filling the spherocylinder uniformly, remember I told you that E. coli cells, we're going to model them as spherocylinders. This gray background sort of represents what the expected distribution should be. And these black points are sort of the profile of the ribosome. And you can see that they're about 4 to 5 times higher where the DNA is not. Like in the DNA-rich region, there is very few ribosomes. The segregation is pretty high. And this is very, very underestimated if you actually looked at an image from a white field image, which is like just looking at all the fluorescence at the same time. On the other hand, if you look at RNAP, the RNA polymerase molecule, I just put 2 kinds of cells, one is short and one long. So you can see that in this case also they are pretty much localized, and when they're long, the DNA segregates and RNAP also goes with those lobes. And if I now take this cell and look at the density of RNAP over this axis, this is how it looks like. And again, the gray area is sort of the expected profile if it was uniformly filled. And therefore, you can see that RNA ribosomes rarely go near the DNA-rich region, and there is almost no RNAP in the end-cap region where ribosomes are most. So with this, we get a first picture of like how they are distributed inside a live cell. We see that RNAPs, if you think of these black lines, this is how I image, like I think of my DNA in the E. coli, DNA in the E. coli. And then you can see the RNAPs are all localized on the DNA, and ribosomes are all outside and in between those 2 lobes. And this is pretty consistent with the idea, with the fact that our lab did a work in collaboration with the Jethiraj lab, where they modeled ribosomes as solid spheres and then DNA as a hyperbranch polymer, and saw that just based on configurational entropy, this hyperbranch polymer DNA stays away from the wall, and the ribosomes sort of fill up the space. So this was sort of consistent with that. So we sort of answered the first question, like why, like how they get localized that way. Probably based on entropy and excluded volume effects, the ribosomes can be localized this way, away from the nucleus. And RNA polymerases, because they're bound to nucleus, they stay localized on the nucleus. Now, what we can also do is that we can estimate actual concentration gradients, concentration profiles. What you can do is that you can look at the intensity from all the molecules in a cell and compare it with a single molecule, and you can get the ratio and calculate how many molecules are there. And we calculated there are 55,000 ribosomes per cell, and there are around only 5,000 RNA polymerases per cell. So after that, and we have the distribution profile, what we can do, we can get a sort of zone-based concentration estimation. And what it gives us is that if you look at the end cap, what we call the ribosome-rich regions, there is 65,000 ribosome per micron cube of volume. That's a very high volume that's occupied by ribosome, and I'm going to talk about the implications of that. On the other hand, in the DNA-rich region, there is around 11,000 ribosomes per micron cube. RNA polymerase, on the other hand, is very few RNAP in the ribosome-rich region, and therefore only 300 RNAP per micron cube. In the DNA-rich region, there is 4,000 RNAP per micron cube of volume. Now, what does this tell us? So, before I started working on all ribosomes and RNA polymerase, what we started doing is that we started tracking just the fluorescent tag itself. It was just simple fluorescent protein, not tagged to any identity. And what we found is that even though the protein seemed to undergo a simple Brownian motion, its diffusion across the entire cytoplasm was not homogeneous. So this is sort of what we call a diffusion map. The colors are here showing you the diffusion constant estimated with that particular region of the cell. And you can see the protein was sort of diffusing fast in this region, which is sort of close to where the DNA should be. And it was slow in the end cap and between those two regions. So if I take this map and sort of average along this axis, you can get a profile like this. It tells you that diffusion is pretty high in the DNA-rich regions and low when the DNAs are not there. And I just told you that there is a high amount of ribosome in those regions whether there is no DNA. And we calculated that actually 22% of the volume in the end cap is occupied by ribosome. And these are big objects, and therefore they can act as crowder and can really slow a diffuser motion. So therefore that sort of gave us a possible rationale for this effect of diffusion constant. On the other hand, there's a electron micrograph study on imaging ribosomes, mRNA and DNA inside cell, and what sort of they found was that all the ribosomes were associated with the mRNA. So if you think of this line here, it's hard to see, is the DNA. From that there is an RNA polymerase sitting that's making an mRNA, and a lot of these black dots are ribosomes, and they're all sitting on it. And so the idea was that probably all the ribosomes are making proteins while the mRNA is being synthesized by an RNA polymerase. And the problem is that if that was true, in NCAP we estimated that there are 150 to 200 ribosomes per RNA polymerase. So if this picture is true in a way that that's all that there is to translation, then therefore we should be able to associate this large number of ribosomes with an RNA polymerase. The problem is that an mRNA on an average length is like 900 nucleotides. So you can probably associate 15 to 20 ribosome with an RNA polymerase. So therefore, we conclude that most ribosomes which are here, if they are translating, so I'll determine that soon, if they are translating, then they cannot translate mRNAs that are just being made by RNA polymerase. They have to translate mRNA that are done transcribed, that are transcribed already and moved to these regions. So that sort of told us that translation majorly is happening on mature mRNAs. Now, so far I talked mostly about what all the ribosome and all the RNA polymerases are doing. But what we can do, because we can study their dynamics, we can sort of look into their subsets of their behavior. For example, if you think of RNA polymerases inside the cell, there can be majorly three classes of them. One kind of RNA polymerases that are, let's imagine this circle as the entire DNA of E. coli. And these sort of spheres are all the RNA polymerases. And then there will be RNA polymerases that are bound to the chromosome and they're making mRNAs and ribosomal RNAs. So we call them transcribing RNA polymerases. And then there are ribosomal RNAPs that are sort of free in the cytoplasm, but they non-specifically interact with the DNA intermediately. And so we can, in our time scale, we see them as a mixture of these two states. And we want to sort of see those states separately. So what we did is that we tracked single molecules, and as we tracked, we immediately saw that there are two kinds of motion. There is RNA polymerases that just don't move around their positions, like this, and then there are RNA polymerases that are pretty fast. So if I look at all the RNA polymerases, here's only probably around 20 of them from a single cell, and plot the trajectories of them, what you see is that there are trajectories like this, where RNA polymerases are not moving much. And then there are trajectories like this, where the RNA polymerases are sampling a lot of volume. So our first intuition was that these tight trajectories are from RNA polymerases that are bound to the DNA and that are making RNA, so they're involved in transcription. And then these fast trajectories are probably representing RNA polymerases that are free in the cytoplasm and probably interacting with the chromosome. So what we did, so René Dalrymple in our lab was tracking positions of a chromosomal foci, which means that she has a label on the DNA, and she's looking at how the DNA fluctuates over time. And so we looked at multiple of these positions, and in general, to compare diffusion constant, what you look at is called mean square displacement. So it scales almost linearly with time. However, this is not a diffusion. In diffusion, it will scale with time linearly. But this is probably subdiffusion, which other people have shown for DNA. So therefore, these plots of mean square displacement sort of curve down. And we saw that how the DNA was fluctuating was pretty similar to how our slow RNAPs were fluctuating. So that was consistent with the fact that those RNA polymerases were bound to the DNA tightly. What about the other fraction? When we modeled the other fraction, we saw that their mean square displacement curves was also sort of bending over in time. And there's again two cases. One is that they are subdiffusing like the DNA, or the fact that they're so fast that they might be hitting the wall. They have some kind of boundary to them that they cannot get away from, and therefore the MHD cannot go as fast, cannot increase as fast as before. And so what we did is that we saw before that RNA polymerases are localized on DNA. So we sort of wanted to look at the size of a DNA loop. And so Wenting Lee in our lab, she imaged HU protein that binds to the DNA, and this is a super resolution image of the nucleus of a cell. And we sort of estimated the size of those nuclei. What we did then is that I took this size and I modeled. So if it can be very well modeled as a cylinder that has a length of 1.2 micron and a diameter of 0.8 micron. And if I model a random walk that is a diffusion constant of 0.2 microns square per second, then this is the black line is what you get. That's a pretty good estimation, sort of representation of what we are seeing in our experiment. There's two kinds of data in this plot. There's blue plot data that is coming from fast tracking at 30 millisecond per frame, and the red data is from 100 millisecond tracking. The 100 millisecond tracking data sort of deviates a little bit away from this plot. It's probably from the fact that at a longer time scale, RNA polymerases can jump from one loop to the other, so it starts to deviate. So therefore, we think that this population represents the mixed state RNAP. They're RNAP that are free, but because of the fact that they're binding to the DNA very frequently, they cannot leave the DNA. So the confinement that we talk about here in the cylinder is not from a physical barrier, but rather is coming from the fact that they're interacting with the DNA, so they cannot leave the DNA for too long. And so with those in our hand, that now we know that there are two kinds of state, we wanted to know where the transcribing RNA polymerases are in the cell. So before that, forgot about this. We also confirmed that if our assignments are right, if those slow RNA polymerases are representing transcription, then over some time, if you look at a long time, those transcribing RNA polymerases should finish transcription and become free. Same thing, vice versa, if the RNA polymerases start seeing as a mixed state, at some point, they should start transcribing and become slow. And so what we did is that we imaged at much slower time scale, so 2 Hz, 500 millisecond per frame, and this is what you see. So this is a movie where an RNA polymerase is transcribing. It's slow initially, and then it finishes up and releases. So consistent with the fact that if the RNA polymerase was bound to a DNA specifically for a long time, it releases sort of around after 5 or 6 seconds of this movie. On the other hand, here's an RNA polymerase that was free initially, but it finds a position and stays there and transcribes. So based on these kind of transitions, we saw and we sort of calculated the expected frequency of these kind of transitions, and compared with our observation, it was consistent that these probably are transcribing RNA polymerases and mixed RNA polymerases. So now, as I mentioned before, that we wanted to look at where the transcription is happening. What we did is that we imaged the DNA. So here's a single cell, and the green signal is coming from a dye that binds to the DNA. And the red dots are sort of the positions of those slow RNA polymerases. And we're looking at where they are on the chromosome. And just looking at these images, it felt that these RNA polymerases are mostly at the peripheries of the nuclei. Remember, this nuclear image, this green one, has a lot of broadening because it has diffraction problem. And so what we did is that we sort of wanted to quantify, and for that we had combined a lot of cell worth of data. We took a lot of these kind of cells, and what we calculated from their phase contrast image is this blue line, which is their center line. You can think of that as the spine of that cell. And we calculated the distances of a lot of RNA polymerases from their corresponding cell center line, and this red curve is the distribution of those distances. Now the green curve is such similar distances, but for the protein HU, that you think probably binds to the DNA uniformly. And you can immediately see that the red curve is much wider, which suggests that probably transcribing RNA polymerases are at the periphery of the nucleus. They are at least more sort of distributed near the surface of the nuclei. Also, there was another interesting finding is that we compared these distributions with, again, what this blue curve here is the distribution of a uniformly filled protein, filling protein. And we saw that their distributions are quite similar near the edge of the cell. So here's it's marking 450 nanometer position, which is the radius that we measured for the average cell. And it goes pretty much out around 100 nanometer or so because of the localization accuracy issues. So what are the conclusions from that? So if we now, again, going back to the original picture of like where RNA polymerases and ribosomes are, we think on an average, transcribing RNA polymerases, these yellow ones, are sort of near the periphery of the cell. And ribosomes can be there that are sort of linked to those mRNAs and doing co-transcription and translation. We still haven't segregated different kind of transcriptions. And probably, based on our comparison of the distribution of transcribing RNA polymerases and all the RNA polymerases, if you subtract that, you get the RNA polymerases that are mixed state, and we think they are uniformly distributed over the chromosomal DNA. And the fact that we find a lot of RNA polymerases very close to the membrane, it suggests that it's consistent with the fact that's called transduction. It's a process when membrane proteins are made from their corresponding genes, and as the mRNA is being made from the gene, ribosome sits on them, makes a protein, and the protein gets simultaneously incorporated in the membrane. And this whole process can pull the DNA close to the, has a potential to at least pull the DNA close to the membrane. So the fact that we see RNA polymerases near the membrane is consistent with this transduction theory. And I'll give another sort of proof that this may be true. So we're done, we are here, we sort of stopped with RNA polymerases and we started thinking about ribosome. And same kind of partitioning problems can be thought with ribosomes. Ribosomes can, generally they are translating these mRNAs and they make the protein, but once they are done making the protein, these ribosomes leave the mRNA and they become free, and then they search for another position and they become bound. So therefore, you can think of roughly two kind of state again, ribosome that are translating, ribosome that are free and searching. And so again, we did single molecule tracking of ribosome. And if you can see probably that you can see that there are really fast molecules and then there are molecules like these that don't move much. And so what we did is that for each cell, a single cell, here are some trajectories of ribosomes, how they are diffusing inside the cell. And you can see as expected, they come only in the end cap and between the two DNA lobes. And what we calculate is distribution of diffusion constant, which is how fast each of those ribosomes was diffusing. Because these trajectories are short, there is an associated statistical noise for the estimation of diffusion constant of a single trajectory. And so therefore, you should expect a broadening. But when you looked at this the observed distribution, which is these red bars, the broadening could not be fit with a single population. However, we could fit it roughly with 65 to 85% of the molecules that are diffusing pretty slow, 0.03 microns per second. And another 15 to 35%, they are diffusing much faster with a 0.12 microns per second diffusion constant. And this was, like, if we think now that these slow molecules, roughly 80% of them are sort of translating, that's consistent with previous biochemical estimate. And because there are estimates saying that roughly 80% of the ribosomes are translating. So based on that, we assign that this fraction is translating ribosome, and this was sort of searching ribosomes, probably interacting also with the mRNA for this process. What we also did is that we then looked at much longer time scale again, tracking at 2 Hz. So this data was taken at 33 Hz, so it's pretty fast tracking. So we wanted to look at if there's a subsection of those ribosomes that are translating, subpopulation. What we saw is that there is a population, I plot them as red trajectories, that are really, really slow. Because now you stretch your time, you can really see who is slow and who is fast. Because if your time is fast, then your localization accuracy becomes close to your movement per frame. So we stretch the time, and we see these red trajectories that are sort of representing very slow ribosomes. Interestingly, we looked at again their distances from the center line of the cell, the spine of the cell, if you remember I mentioned before. And we found, so the green curve is sort of the observed distribution of those slow ribosomes. Now if you compare that with this blue distribution again, if those ribosomes there are filling the cylinder uniformly, you see that it's really different. On the other hand, if I compare that distributions, this red with the red line, which is sort of what you expect if something was bound to the membrane, then there is a pretty close similarity. And we think these deviations near the peak and the tail region is coming from the fact that this data acquisition takes a long time because it's a slow tracking data, and therefore the cell can drift because of thermal fluctuations. So it's possible that during our acquisitions, our center line assignments are not right. So we have to sort of repeat this experiment with a way that we can keep track of the cell's image as we take the data. So that will be much cleaner estimate of this kind of distributions. So we think that now these really slow ribosomes that are very close to the membrane probably are representing ribosomes that are involved in transaction, that are involved in making proteins that are inserted into the membrane as they are being produced. So with that, our final picture becomes like this, is that RNA polymers are on the nuclei, and they're probably making mRNAs and ribosomal RNAs, and ribosomes that we found around 10% of them are near the nuclei. They can be involved in co-transcriptional translation, and majorly, like 85% of the ribosomes that are in the end caps, they're probably involved in translating those mRNAs that are done transcribing. So mRNAs have been synthesized, they diffuse here, and those ribosomes then sit on them and make proteins out of it. On the other hand, we found convincing evidence that there are RNAPs and ribosomes that are near the membrane, which can be candidates for transduction, where RNA polymerases are making mRNAs for membrane proteins, ribosomes are associating with them and inserting those proteins to the membrane. So that's basically the findings of my graduate work. With that, I'd like to thank people now. I'm really, I'll first acknowledge my advisor, Professor James Weiser. I mean, his encouragement and advice has been one of the key things for all that I have achieved in this graduate school. I'm very thankful to my committee member. Professor Rick Gorse has been in my committee from my thesis background oral and research proposal. And I'm thankful to Tom, Jade, and Sylvia for coming to be in my committee with such a short notice. I'm thankful to Professor Mark Bullion from UPenn for providing those strains that I use for ribosomes, and also he has provided me with great advices over the time. Thankful to Tamas Gal from Gorse Lab for providing a lot of strains that are useful in our study. Rene has also used some, and I have used RNA polymers from him. Suparna Sennal from Uppsala University has provided us with the ribosome strain that we are using right now. And I'm thankful to my group. This is a picture of our group, and they've been very supportive and very helpful over the career. And our past members, I couldn't put their picture, but I learned a lot from them too. But other than the group and the science part, there have been a lot of people inside and outside the department that have helped me and sort of been a big part of my life in medicine. And I just want to name some of them, but my whole batch of the chemistry department and other people, I cannot thank all of them, but they are really, I'm really grateful to them. So, but the people, like I really want to mention Jagannath. He was my senior from the monastery that I mentioned. So I'm sort of following Jagannath, tracking him down over up to here. But he has been a great mentor, and sort of he encouraged me in research a lot and stuff. Shudipto was my roommate. He just graduated this summer. We had great discussions when we were roommate, and it was a great time with him. Pooja and Kwang are one of the two people in my batch that have been my great friends, but all the other people are really great friends too. Jayashree and Arnav were also two people. One of them was from a mathematics department, and Jayashree was a student in Nate's lab. And I had really good time discussing science and other photography and things with them. And finally, I want to thank E. coli for taking all the research doses and letting us to see inside them and sort of helping us to know biology. I really want to thank my family members. These are my parents, my sisters, my grandmother, and my other family members. This is near the airport. Whenever I used to go, there's like 20 people waiting for me. And yeah, there's a big picture there. But these are my friends from my high school, my IIT Kanpur, my college, and I still have very great relationship with them. And whenever I'm not doing science, and I have always a place to go back to. So that feels really great. And thank you all for your attention today. I'll be happy to answer any questions.
Based on the blue slower, do you think you could infer that there are more ribosomes found?
So we can, most of the time when you're looking at these kind of phenomena, we have a sense of average picture. So we can probably get a sense of how many ribosomes on an average are sitting on an mRNA. For example, if we quantify what fraction of ribosomes are translating and the fact that we know the total number of ribosomes, and then you can calculate like how many mRNAs are there by some other people's study, or we can compare and see like how many on average are sitting. However, these large number of systems, the fact that there are 55,000 ribosomes makes it very difficult to do a colocalization study. For example, like you cannot see like 5 ribosomes at the same time or something to colocalize them. So it's very stochastic, and we look at average picture. So indirectly, we can sort of get we can sort of infer like how many ribosomes per mRNA, but to sort of directly see that will be quite difficult.
What's the answer?
What's the answer? Question, but I think we estimated it to be around 8 to 10 ribosomes per mRNA. Yeah, but that's based on the fact that also that mRNAs, a lot of ribosomes are sitting on mRNA that are not synthesized completely. So there are mRNAs that are short and being produced. So therefore, it's a big average over a lot of unequal numbers.
Other questions? Come on.
There's question, Cornelia.
So we were like showing us, you know, the RNA.
And polymerase?
Yeah.
Okay.
Like, you know, it was like docked onto the RNA and then we leave. So do you have an idea how fast this transcription process happens?
So we are trying to do that kind of measurement. So biochemical assays have provided good estimations in that way, like how fast an RNA polymerase makes an mRNA. And so like so this, if I go back to sort of the original picture of, so when an RNA polymerase is making mRNA, they're making it base by base, and people have very good estimation that how fast it makes it. Apparently 3-4 base for an mRNA, an RNA polymerase can make it. And we're trying to, so those are biochemical assays, and we're trying to get an estimation of those from also our in vivo assays in live cell. The way to do that would be, we're thinking that if we can look at how long on an average an RNA polymerase stays like a stuck RNAP, and then it releases, and if we can get, again, like I explained, that if we can get an estimation of average mRNA length and compare the average residency time of a slow RNA polymerase, we can sort of get a rough estimation of how fast it's doing transcription. Okay.
So it sounds like this isn't necessarily your work, but it's more maybe someone else's in your group. But can you talk at all about how the DNA loci that you're shining and which that's good.
Yeah.
So and whether some of those loci are in the maybe in the middle of the lobes versus on the outside of the lobes where they might be more transcribed.
Yeah, it's a good question. I mean, it's a very valid point, and so people have shown that for a lot of different kind of foci, for example, for membrane proteins, for example, the genes sort of move when you are transcribing them. So it's, yeah, we haven't sort of looked at the sort of heterogeneity, probably if I'm right representing Rene here, that we haven't looked at the heterogeneity over like how different foci are distributed, like diffusing together, and we should be, yeah, we can look into those kind of things. That's a good point, that yeah, it's true that when they're being transcribed and when they're free, they can have a different kind of dynamics and even positions too.
Other questions? Well, thank you for coming.